give a talk on identification of Phragmites lineages in your region. And I'm also recently back from a symposium that was held on Phragmites at the Society of Wetland Scientists the first week of June. And we had quite a few discussions about Phragmites management at this meeting, so I'm going to be talking a bit about some of the things that we covered there as well. So with that, I will get started. I'm assuming that you all are familiar with Phragmites. I'm not going to talk a bit at all about its basic ecology, where we find it, those kinds of things, because I assume you're all familiar with it. Here you can see some maps of the distribution of the 3 major Phragmites lineages that we have in North America. On the left, you see native Phragmites australis, which we now call subspecies americanus. The historical record shows that this subspecies has been in North America for thousands of years. We have records from the Southwest that go back to the Pleistocene. In New England, you can core in the salt marshes and find rhizomes that are 3,000 to 4,000 years old. So we're very confident that Phragmites is a native part of the flora in North America. The middle panel shows the Gulf Coast subspecies, which is called Berlandieri. This lineage is represented by a single chloroplast haplotype, which is haplotype I, and its range extends down through Central America into northern South America. So this is a hexaploid lineage. It's quite different from the Phragmites we have up in the north and the northeast, and it's restricted to the southernmost states. It has been introduced into Arizona and Southern California, and it can be aggressive in these places. And then on the right, you see the map of introduced Phragmites australis, which we think was likely introduced to North America sometime in the early 1800s. It likely came across from Europe as ballast material on ships, because our earliest herbarium records come from ballast grounds along the Atlantic coast. It started spreading into the marshes, and has spread west since then, and we now find it everywhere, just about everywhere in North America, up into southern Canada. We don't know the extent of its range down into Mexico, and if it has made it farther south, it may be that this is a temperate lineage that can't exist in the tropics where I live. But it certainly has become widely distributed across North America and is responsible for the invasions we see. Is someone talking to me? No. Okay. So I'm going to start talking about the morphological differences between the native subspecies americanus and the introduced Phragmites. And this is a photo of me in my younger days at Montezuma National Wildlife Refuge in Seneca Falls, New York. The Phragmites you see on the right that I'm looking at is native Phragmites, and the patch on the left, the darker one, is introduced Phragmites. So I'm going to start talking about the morphological characters that I like to use to distinguish the 2 lineages. Some are better than others. None are entirely accurate in and of themselves, so I want you to keep that in mind as we go. There are some that certainly I look for first, and these are the ones that I'm going to be emphasizing. So the first thing that I look at when I go to a Phragmites stand is I look at the community as a whole. Is this a monoculture, or are there other plants growing there? So typically with native Phragmites, we find it growing in mixed communities. The stem density may vary. It can be quite dense, it can be very sparse, but we usually find it growing with other plants. In contrast to introduced Phragmites, which, as you all know, typically grows in dense monocultures. If we see other plants, it's usually only along the edges. Often these are vines that are growing up the stems. Occasionally, you find stands that have low density. This is usually because they're very poor quality sites or because they're young, newly establishing stands. So the first pass I look at is just getting a general sense of the community. But then getting into the individual characters. My favorite character is to look at the ligule height. So the ligule is a membrane that you find at the base of the leaf. It's on the inner surface where the leaf is wrapping around the stem. You can see the arrows pointing at it. in these examples, these come from herbarium specimens, so these are dried specimens. But the ligules are the brown membrane that are being pointed at. And you can see that the native one, as you see on the left, is quite a bit larger than the introduced one. There's a fringe of hairs that's, that extends upward from the membrane. This fringe can also be measured. There's often some very long hairs. These break off easily. We don't include these in the measurements. But really the membrane itself is what we need to look at. This next slide shows the same thing with fresh tissue. Again, you can see the membrane on the left for the native is quite a bit bigger than the introduced one on the right, where it's very hard to see the membrane itself. These photos were taken with a dissecting microscope. That's usually what you need to look at to really get a good measurement of the ligule size. But if you have examples of a native and an introduced ligule side by side, you can see with the naked eye that there are distinct differences in the height of the ligule. Moving on, the next character I like to look at are the glume lengths. For this, you need an inflorescence. It can be an old one from previous years, or it can be a new one. So the glumes are found at the base of where you find the fluffy seeds in the Phragmites panicle. On the left, you can see an example of the seeds. And on the right are the glumes themselves. There are two of them, a lower and upper one. The lower ones are the ones that are seen on the left in these photos. And you can see from the photo that the introduced glumes tend to be, again, much smaller than those we see in the native. Now, I've been measuring glumes now from samples collected all over North America, and there is a little bit of overlap. But typically, the native ones are larger. Most of them are greater than 4 millimeters in length, and the introduced ones are smaller than 4 millimeters in length. Most of them are range about 3 to 3.5 millimeters. So this is a character that you can look at in the field. If you have a little ruler with you, you can get a good idea of the glumes. You typically measure 4 to 5 per stem. Take a few stems, takes a few minutes, but you can get a pretty good idea of the average glume length within a stand very quickly. The next thing to look at is stem coloration. Although this can be a very subjective character, some people see colors differently, and there also is some overlap in this character. But typically, introduced stems are green, and native stems may have some reddish purple color on them, or they may look yellow. And this is often because the leaf sheaths, the base of the leaves that wrap around the stem, fall off much more easily in the native plants than they do in introduced plants. So where the stem is exposed to the sun, it changes color. I just mentioned that the leaf sheaths tend to stay more tightly bound to the stem in introduced plants. These are dead stems that you see here. On the left are natives, on the right are introduced culms. And as you can see on the left, most of the leaf sheaths are missing or very loosely attached to these overwintering culms, whereas on the introduced examples on the right, nearly all the leaf sheaths are present in overwintering culms. So this is a character that extends throughout the year. During the growing season, the lower stems of the native plants will lose their leaf sheaths, and the introduced ones will hold them, and this persists throughout the winter after the stems have died back. And here's an example in live plants. On the left, again, the native. My arrows got cut off, but you can see the leaf sheath has opened up on the native stem. The leaf has actually died back. This is down at the base of the stem. And there's a lot of red color there where the stem has been exposed. On the right, we see an introduced stem. The leaf sheath is tightly wrapped around the stem. There's only the green color that comes from the leaf itself. And just looking at the stand as a whole, you can really see this color variation when you look down below the area where you have a lot of green leaves. In the native stands, you will often see a lot of variation in stem color. You'll still see some green stems, but you'll also see a lot of red color, whereas in introduced stands, you see green throughout the whole height of the stand. Even down below where you have dead leaves, where there's not much sunlight getting down there, it just looks like dead tissue. You don't see any reddish color for the most part. So the stems stay green down the length of their stems. Another thing to look at on the stem is the texture. Introduced stems tend to have ridges on them. They have a flat color to them, whereas native stems are shiny and often very smooth. You can feel this difference by running your thumbnail along the stem itself. To do this, you often have to take the leaf sheaths off, so you need to unwrap them, and then you can feel the stem itself. We also see some fungal patterns where introduced stems will often have sort of a mildewy looking fungus growing on them. Native ones can get these nice black spots growing on them. This is a fungus. I don't know what it is, but to my knowledge, these spots have never been seen on introduced stems. Not all natives will have them, but if you do see them, it's a good sign that it's a native plant. And here's an example of a dead native stem taken in the wintertime. You can see very clearly this pattern of black spots underneath where the leaf sheath had wrapped around the leaf when the plant was still alive. Okay, another thing to look at are leaf color and the angle of the leaves coming off the stems. On the left, you see native plants. On the right are introduced ones. The native leaves tend to have more of a horizontal angle coming off the stem. They tend to be a bit floppier, whereas introduced leaves tend to be stiffer. They come off at a more of a 45 to 60 degree angle off the stem. This is a fairly young plant that we're looking at here. They do, as the leaves grow, they get heavier, they flop down a bit more. But you can see differences in the angle of the leaves coming off the stems. There's also some differences in their colors. Native plants often have a more yellowish green color, whereas introduced plants have a darker bluish green color. Again, this is a very subjective character. It's when you see the two different types of plants side by side that you can really say, okay, yeah, I see a difference here. As here, you can see here in this photo, again, on the left we have introduced Phragmites, on the right is native, and you can clearly see that the bluish green color on the left is more indicative of the introduced, whereas the native plants are a lighter yellow-green color. In the fall, we can also look at timing of senescence. Introduced plants typically stay green later in the year than native plants. Native plants can also flower earlier, although there certainly is a lot of overlap in the timing of flowering. But at least along the Atlantic coast, where I've done a lot of looking around at native plants, they typically start senescing in late August, early September even, whereas the introduced plants will stay green until October, even into early November in a mild year. So that's a good time of year to go out and look for native Phragmites because it typically is brown, whereas the introduced plants are often green still. And we can see these differences from the air. This is an aerial photo that was taken on the Delmarva Peninsula. I don't know if you can see my cursor here, but I'm pointing at some patches of Phragmites. Over on the left, there's a nice patch of grayish-looking stuff next to the river. That's introduced Phragmites. There's also some patches in the very middle of the screen. Uh-oh. Well, okay, here come the delineations. What the green ones are coming individually. These are all native stands that I'm picking out here. And here comes the introduced ones. Okay, sorry there. I didn't realize there were so many here. On my computer, they were all coming up together. So you can see here that we have a nice cluster of native Phragmites populations in the foreground, but in the distance, it's all introduced Phragmites. And if you have good photos like this, you can start to delineate stands from the air, which can help us find native populations as well. Okay, so the best characters that I've just gone over. My number one character is ligial height. I also like to look at the gloom lengths. These are two characters you can actually measure. You can write them down, keep a record of them. They're measurements, so they're not subjective at all. Adherence of the leaf sheaths to the dead stems is also a very good character. As I said, the native ones will tend to lose these leaf sheaths, whereas introduced ones hold onto them throughout the entire year. Then we also look at a combination of other factors. We look for the presence of the fungal stem spots that are indicative of natives. We look at the stem texture, whether they're shiny and smooth or ridged and more dull looking. Shiny and smooth is native, ridged is introduced. We look at the stem color. Is there any reddish color on the stem? Is this reddish color extensive? Is it found across the stand? That's indicative of native. I already mentioned the stem texture, shiny or ridged. We look at the stand characteristics in general. Is it a monoculture? Is it a mixed community? Is the stem density really high or is it lower? Again, this can be related to the stand age, but we all know that dense monocultures are typically introduced Phragmites. Another thing you can look at is the herbivore community. I have not looked at this at all. This is far beyond my expertise, so I didn't want to really talk specifically about it. But if you're interested in differences in the herbivore community, I suggest you get in touch with Dr. Bernd Blasi at Cornell University. He's done a lot of work surveying Phragmites populations for their herbivores, and he can tell you more about what to look for. So I mentioned again that there are a lot of characters that overlap though. So stem color is a very important thing. Both introduced and native stems can have red color, especially at the nodes. This is a photo of introduced Phragmites that was taken in southern Connecticut. You can see very clearly that there's red color on the stem, and if you look closely in the background, you can see it on other stems as well. I mentioned stem density. Both stands can be sparse or very dense. So you need to be careful looking at that. It's the community as a whole you look at. Stem height is not a good character. You can have very short introduced Phragmites, very tall native Phragmites, so that's not one that should be used at all. The size and shape of the panicle, the inflorescence, is also not a good character. Another one that's been talked about is the color of the leaf collar. The leaf collar is at the outer edge of the leaf at the base where it connects to the stem. Some have said that red color is indicative of natives, green is indicative of introduced. This is not true. There's also overlap in this character, and it should not be used as a way of distinguishing the two lineages. So basically, when you're making these calls, you need to think about, does anything about the population look introduced? What I do, if I have any question in my mind, is I go and find a roadside population and do a comparison. And if you find anything that looks introduced, chances are it probably is introduced. So you need to keep that in mind and not get excited too easily about the possibility of finding a native. So there are several ways that you can make sure. There's genetic tests. There's an RFLP diagnostic test, which is a fairly simple genetic test that can be done in a laboratory. I used it in for students. A lot of people are using it that I know for student laboratory methods. I'm happy to send out protocols to do this. This will tell you whether you have a native haplotype or an introduced one by running a simple agarose gel. You can also do sequencing, or you can look at nuclear DNA and look at microsatellite analyses. Anyone who's interested, please get in touch with me, and I can provide you with protocols and information on how to get this type of work done. So, but recently, as you've probably heard, Phragmites hybrids are starting to be found. These are hybrids between native and introduced Phragmites. And as you may have heard, these have been found at Montezuma National Wildlife Refuge in Seneca Falls. So I'm not going to show you any photos of these hybrids, but the ones that were found there, they look more like native plants than introduced ones. They have long ligules, they have glossy stems. The leaf sheaths remained on the overwintering stems, but they were loosely attached and didn't really have a firm connection. So that they were sort of an intermediate character there. But another thing that convinced us that these may be hybrids was the pattern of insect attack. Again, I haven't talked to you about this, but Bernd Blasi is a person who can talk to you about this more if you're interested in the herbivore communities. We found 3 hybrid clones at Montezuma. They were all identical, and they were all located near the entrance to the refuge, so we speculate that they were probably spread by landscaping-related management activities. We know very little about the reproductive ability of these clones or how likely they are to spread, but in my opinion, these clones should be eliminated. They should not be left there to see what happens. So we're starting to find Phragmites at multiple sites around North America. They've also been found in Maine. They've been found in Virginia. Some suspected ones have been found in Massachusetts. These need to be confirmed. And we've also found extensive incidences of hybrids out in Las Vegas, which is a whole other story I'm not going to talk about today. But hybrid Phragmites is starting to be found. In general, we find these hybrids at sites where both native and introduced Phragmites co-occur. They're very small clones at this point, but again, we don't know how quickly they might spread. Morphologically, they're very difficult to distinguish, and you really need genetic tests to confirm identity. The best tests to use for this are microsatellite markers. This is a graph here that's based on 8 different microsatellite markers which look at nuclear DNA, so you can detect both the parental lineages. And you see what on the left is a cluster of introduced samples. On the right are native ones, and then down in the middle is the hybrid individual. This is the example from Montezuma. So you can distinguish very clearly between the 2 lineages and their hybrids using this technique. Okay, so I'm running out of time, so I'm quickly going to go into some of the management things that we've talked about. Again, I just want to say a few things about introduced Phragmites in general. We now find it across North America. It's very opportunistic. It thrives in disturbed habitats. We know it likes high nutrient conditions. It can also tolerate high salinity, but it loves freshwater sites, so it really can grow just about anywhere. We also know that historical management practices have likely facilitated establishment and spread. So we find it along transportation corridors. We find it where lowlands have been drained for conversion to agriculture, where there's a high associated nutrient runoff, where shorelines have been developed for housing. There's high nutrients associated with this as well. And we also find it along river systems where flooding can disperse rhizomes. It spread generally occurs by seed. The seeds are when dispersed. They can be very, seed viability can be very high, especially in areas where there's extensive Phragmites stands. So these are things that certainly have facilitated its spread. And here's a brief overview of some of the management things we talked about. There's a paper that recently came out in 2014 by Eric Hazelton and colleagues looking at Phragmites management in the United States. They looked at 40 years of studies that have been done. This actually turned out to only be 34 studies that were published. But this paper is publicly available. I have the website there for you if you're interested in downloading it. But they've done a very thorough overview of what we know from the literature about these things. And what they found was that most of these studies have been very short-term. Herbicides, cutting and mowing are the most common methods that are used for removal. But most studies have focused on individual wetlands and small patches of Phragmites in a relatively small area. So we really have very little information on the success of much of the management that's been done on community-level recovery of vegetation and across a landscape. So none of these studies have really been placed in a landscape context. And in my opinion, and in the opinion of much of the people at this meeting, that is really where we need to go with Phragmites management. We need to think about it on a large scale. Disturbance is widespread. Opportunities for seedling establishment are everywhere. Nutrient inputs are everywhere, so eutrophication is widespread. We know seeds are widely dispersed. The wind blows them everywhere. Machinery disperses them around. There's many ways for seeds to move around. So one of the things we need to think about in a management context is removing seed sources as much as possible. Following removal, we need to make sure that we prevent reestablishment, so long-term monitoring and future treatment is always important to keep in a management plan. And revegetation may be needed as well if native plants don't establish. So work has shown that in the Northeast at least, the native seed bank is often quite high in Phragmites marshes, so following removal, native plants often establish themselves quickly, particularly when there's nearby wetlands with native vegetation. But management is a social process as well as a biological one. So we need coordination across landowners. Of course, this is a mix of private and public landowners. Goals need to be specified, and efforts need to be prioritized. We need restoration of native communities may be very different than thinking about habitat for wildlife, or if we're trying to restore ecological function of the system. So efforts need to be prioritized to maximize treatment success, and in some cases, it needs to be recognized that if infestations are extensive, eradication may be impossible. So newly invaded sites with small stands should be given priority. Seed production and dispersal has to be dealt with to prevent further invasion, so timing of treatment to prevent flowering is very important. And where preservation of native communities is a management goal, we need to remove all introduced Phragmites to minimize opportunities for hybridization with natives, because now we know hybridization does occur, and we want to prevent establishment of these hybrid stands as well as back crossing into native populations to help preserve their genetic integrity. So I feel if hybrids are detected, they should be removed immediately. If anyone suspects they have a hybrid plant, it should be sent out for genetic testing to confirm its status. But if removal is not possible, you can mow plants down before flowering initiates to prevent further crosses or back crossing and dispersal of seeds, and it's very important to remove and destroy all cut stems to prevent spread via stem sprouts. So I've just gone over a lot of information, and I've run out of time, so I'm going to stop here. But I'm just listing a few resources here for you. The National Park Service has put together a fact sheet. You can download a document from this website which has all the diagnostic characters I just talked about with photos, information about sizes of the ligules and the glooms that you need to look for. This was put together by myself and Joe Swearingen from the National Park Service. Bernd Blassie at Cornell also has a diagnostic service where if you send him plants, he will look at them and give you an idea of whether it's a native or an introduced plant. His website also has a lot of these characters with photos and information about them. And here are a couple of the papers that I've mentioned. The top one is the publication that talks about the hybrid that was found at Montezuma, and the bottom one is the one I mentioned that is about management practices and some of the conclusions and summaries that have come out of looking at all of these studies. And with that, I'll say thank you and happy hunting in your quest for native Phragmites.